Inside a freezing, dimly lit basement lab beneath New Zealand's Te Papa Museum, a massive colossal squid floats in a formalin tank. Its tentacles are curled. Its eyes, the size of dinner plates, stare through the thick glass. For centuries, the Kraken was just a sailor's myth. Then, we found this. Now, a monitor on the wall displays cascading lines of genetic code. Scientists finally cracked its DNA, but the results didn't just map a squid. They revealed a neural architecture so complex it changes what this creature actually is. The dumb sea monster story is collapsing. For generations, the Kraken occupied a specific space in the human imagination. It was the ultimate oceanic terror. Old woodcut engravings from the 18th century show massive tentacles erupting from black water, wrapping around wooden hulls, dragging screaming sailors into the abyss. The Norwegian bishop Erik Pontoppidan wrote about it in 1755, describing a creature so large it was often mistaken for an island. He claimed its back was covered in spikes, its arms could pull the largest warships under, and its sheer size defied comprehension. These stories were treated as folklore, exaggerated tales from men who spent too many months staring at empty horizons. Then the modern era arrived, and science stepped in to correct the record. Biologists identified the real animals behind the legend. The giant squid, known as Architeuthis, and the even larger colossal squid, Mesinicoteuthis hamiltoni. The official explanation was deeply reassuring. These were not monsters. They were just large, sluggish, cold-blooded invertebrates. Science framed them as biological curiosities, simple-minded floating meat traps that drifted in the deep ocean, occasionally getting tangled in deep-sea trawls by accident. Textbooks stripped away the mythology. The Kraken was downgraded from a cunning predator to a passive, almost brainless piece of flesh. That became the accepted version. A giant squid was basically a giant piece of sushi that happened to have tentacles. It lacked the intelligence to hunt deliberately. It lacked the nervous system to process complex information. It was just a big, dumb mollusk. That is what the world was taught to believe. And for a long time, the physical evidence seemed to support it. Almost every giant squid ever recovered was found dead or dying. They washed up on beaches in terrible condition. They came up in fishing nets, mangled and half digested. They looked like pathetic, broken creatures, completely incapable of the terrifying feats described in the old logs. The consensus was settled. The myth was debunked. The monster was dead. But then you walk into that basement lab in New Zealand, and you stand in front of the tank, and the accepted story suddenly feels incomplete. The specimen suspended in the formalin is not a pathetic blob. It is a heavily armed predator. The colossal squid is not just big, it is engineered for violence. Unlike the giant squid, which has smooth suction cups lined with tiny serrated rings of chitin, the colossal squid has massive rotating hooks. These are not static points. Each hook is seated on a muscular stalk, allowing it to swivel 360 degrees. When the squid strikes, the hooks lock forward. When the prey struggles, the hooks rotate backward to hold the flesh in place without tearing free. Its tentacle clubs are dense with these swiveling mechanisms, transforming them into biological grappling hooks. Additionally, the colossal squid possesses highly developed statocysts, the fluid-filled balance organs located near the base of the brain. In cephalopods, statocysts process gravity and acceleration data, giving the animal a precise sense of its orientation in a three-dimensional water column with no visual landmarks. The sheer size and complexity of the colossal squid's statocysts imply an extreme sensitivity to movement and vibration. Its beak, made of dark, hard keratin, is the size of a parrot's, designed to slice through flesh and bone. And then there is the eye. This is the first physical anomaly that refuses to fit the dumb mollusk narrative. The colossal squid has the largest eyes in the animal kingdom. They are the size of dinner plates. In the dim light of the museum lab, the preserved eye stares out, dense and opaque. Evolution does not build hardware like this for a passive drifter. An eye that massive requires an enormous amount of biological energy to maintain. It requires a massive optic nerve. It requires a visual cortex capable of processing staggering amounts of low-light data in an environment where there is no light at all. If the colossal squid was just a slow, blind piece of meat floating in the dark, waiting to bump into a meal, it would not need eyes like that. The eye implies tracking. It implies calculation. It implies a brain powerful enough to interpret complex visual information in real time. But the official scientific consensus maintained that its brain was simple. A donut-shaped mass of neurons wrapping around its esophagus, no different from any other squid, just slightly larger. The anatomy said predator, the textbook said idiot. That contradiction sat unresolved for decades, hidden behind the glass of museum display cases around the world, while the deep ocean kept its secrets. To understand why it took so long to pull the truth out of the dark, you have to go back to the moment the scientific community actually got its hands on a perfect specimen. The story of the Te-Papa colossal squid begins in February 2007 in the freezing, violent waters of the Ross Sea off Antarctica. A New Zealand fishing vessel was hauling a long line through the black water, targeting Antarctic toothfish. The winch strained. The crew knew something massive was on the line. They assumed it was a heavy block of ice or a tangle of debris. When the net broke the surface, they found something else entirely. A colossal squid. Still alive. Still fighting. It was not a mangled corpse. It was a living weapon, wrapping its hooked tentacles around the deck equipment. The crew managed to secure it, but the creature was dying. The pressure change from the deep ocean to the surface was fatal. They hauled it aboard, froze it solid in the ship's hold, and transported it back to New Zealand. It was a global sensation. Finally, science had an intact Kraken, but almost immediately, the investigation hit a wall. The creature's body chemistry was actively fighting the researchers. Squid tissue is notoriously difficult to preserve. Unlike fish, squid rely on a high concentration of ammonia in their bodies to maintain buoyancy in the deep ocean. When the animal dies, that ammonia does not just disappear. It permeates the muscle tissue, effectively acting as a slow chemical poison that degrades DNA. Scientists trying to extract genetic material from dead giant and colossal squid found that the code was fragmenting before they could even read it. The locked room mystery was not about finding the creature. It was about the creature destroying its own evidence from the inside out. This biological self-destruction created a cloud of suspicion around everything we thought we knew. Early genetic tests on degraded specimens gave wildly contradictory results. Different labs testing different tissue samples kept coming up with different numbers of species. Some researchers argued there was only one species of giant squid. Others claimed the genetic data proved there were 3, maybe 4, possibly more. The results were a mess. Tissue degradation and contamination made the early DNA sequences unreliable. The taxonomy of the deep sea was collapsing into confusion. Early scientists looking at dead specimens had already made the same mistake. In the 19th century, researchers examined giant squid of different sizes and incorrectly classified them as entirely different species. They did not realize they were just looking at juveniles and adults of the same animal. Now, modern genetics was repeating the error, not because of bad science, but because the squid's ammonia-laced flesh was destroying the data. And then there was the deeper, more unsettling open loop. If these creatures were so abundant that they kept getting caught in nets, why did no one know how they lived? The bizarre historical fact is that no human being photographed a live giant squid in its natural habitat until 2004. No video footage of a living giant or colossal squid existed until 2012. Consider the sheer scale of that observation gap. By the mid-20th century, humanity had nuclear submarines navigating the deep ocean basins. We had military sonar pinging off the seafloor. Research submersibles like the DSV Alvin, first deployed in 1964, were diving to depths of thousands of meters. We mapped the ocean floor using satellite altimetry, yet the largest invertebrate on Earth remained entirely unobserved in the wild for the entire span of modern marine exploration. The water column where these animals live, the mesopelagic and bathypelagic zones between 300 and 1,000 meters down, is a vast, lightless void characterized by extreme pressure and near-freezing temperatures. It is the largest habitat on Earth by volume, yet it remains the least observed. When Japanese researcher Tsunami Kubodera finally captured the first photographs of a live giant squid in 2004, it was not in some unexplored trench. It was off the Ogasawara Islands, a well-documented region. The creature was not a passive drifter. It attacked a bait line, aggressively snagging a tentacle. 8 years later, in 2012, Kubodera and an American team finally filmed a giant squid swimming in the deep water near the Ogasawara Islands. The video shattered the passive mollusk narrative visually. The squid was vibrant, silver and gold, actively hunting, using its massive eyes to track the bait in the absolute blackness. It did not drift. It maneuvered with precise, jet-propelled thrusts, coordinating 8 arms and 2 feeding tentacles in a highly controlled assault. The 2004 photographs and the 2012 video did not just capture an animal. They captured the failure of 20th century marine science to account for a highly evasive, sentient predator in its own environment. Submarines are loud, churning water, generating heat, and electromagnetic fields. If the giant squid possessed the sensory sophistication its anatomy hinted at, it likely detected these machines long before they detected it. The creature was not just hiding in the dark. It was tracking our technology and avoiding it. The mystery was locked in the dark, sealed inside flesh that chemically refused to give up its secrets. It took years of painstaking laboratory work to finally crack the chemical defenses of the Kraken. The breakthrough did not come from a single dramatic expedition. It came from methodical, frustrating extraction work in sterile laboratories, slowly piecing together fragmented DNA sequences from multiple degraded specimens. The turning point arrived in 2020. A team of researchers led by Caroline Albertin at the University of Copenhagen published the first high-quality genome sequence of the giant squid. It was a monumental achievement in marine biology. They had finally forced the creature's genetic code onto a computer screen. The scientific community expected the genome to confirm the existing consensus. They expected to see a standard mollusk blueprint, perhaps with some size-related mutations, but fundamentally simple, they were wrong. The moment the sequence aligned on the monitor, the data showed an anomaly. The researchers started looking at specific gene families, the instructions that tell a body how to build its nervous system. They focused on a class of genes called protocadherins, specifically clustered protocadherins. This is where the science gets critical, and this is where the dumb mollusk story begins to shatter. Protocadherins are neural wiring genes. They work by randomly combining in individual neurons, giving each nerve cell a unique molecular identity tag. They act like a sorting system in the developing brain, allowing neurons to connect in highly specific, complex patterns. They are the difference between a simple bundle of nerves and a brain capable of advanced processing. In a standard invertebrate, like a normal squid or an octopus, the number of protocadherin genes is relatively low. The California two-spot octopus, long considered highly intelligent for a mollusk, possesses a modest array compared to what was about to be revealed. They have enough to run a basic nervous system, enough to control tentacles and process simple sensory input. In vertebrates, in mammals, in humans, the protocadherin family underwent a massive expansion. We have over 50 of these genes. It is a core reason we have complex brains. When the researchers scrolled through the giant squid genome, they found something that should not have been there. The giant squid had undergone a massive, independent expansion of its protocadherin genes. The numbers were staggering. The expansion rivaled what is seen in vertebrates. The creature drifting in the dark ocean, the animal textbooks called a simple meat trap, possessed a genetic toolkit for neural complexity that biologists had never expected in an invertebrate. The visual comparison chart told the whole story. Standard squid DNA showed a flat line of basic neural genes. Human DNA showed a massive spike. And there, right next to the human spike, was the giant squid, a separate, alien, evolutionary path that led to the exact same biological destination: high-level neural wiring. But the protocadherins were only the first layer of the reversal. The genome scan revealed something else, something that made the neural complexity even more unsettling. The researchers found an explosive proliferation of transposons. Transposons are often called jumping genes. They are sequences of DNA that can copy themselves and paste themselves into different parts of the genome. They are usually considered parasitic or junk DNA, noise in the system. But in recent years, evolutionary biologists have realized that transposons are not just noise, they are a primary engine of evolutionary innovation. When transposons jump, they can duplicate entire gene families, shuffle existing genetic networks, and create entirely new biological capabilities. The giant squid genome was absolutely flooded with them. The jumping genes had not just mutated a few instructions, they had rewritten massive sections of the creature's developmental code. The sheer volume of transposon activity in the giant squid suggested a genome in hyperdrive, actively reshaping its own nervous system over millions of years. This was not a static, simple creature. This was an animal locked in an intense, ongoing, evolutionary arms race to build a better brain. The math of its brain development no longer matched the narrative of a passive drifter. The genetic evidence pointed toward a highly specialized, deeply adapted deep-sea predator. The anatomy was starting to make sense. The massive dinner plate eyes were not evolutionary leftovers. They were the primary input devices for a massively parallel neural processor. The rotating hooks on the colossal squid's tentacles were not random mutations. They were the physical weapons controlled by a brain capable of complex, three-dimensional spatial tracking in total darkness. The old textbooks were wrong. The 20th century scientific consensus had fundamentally misjudged the creature. It was not a dumb piece of meat. It was a biological supercomputer hiding in the abyss. Once the genetic reversal landed, the historical records had to be reevaluated. The implications rippled outward, changing the context of everything we thought we knew. Look back at Eric Pontoppidin's 1,755 descriptions. Look back at the old sailor logs describing ships being dragged backward. Look at the physical evidence left on the bodies of sperm whales. Sperm whales are the only known natural predator of the giant squid. When whales are found with giant squid sucker marks on their skin, the assumption has always been that the squid was the victim, fighting desperately but hopelessly as it was eaten alive. But the scars tell a more complicated, more strategic story. Marine biologists examining stranded sperm whales have noted a specific, recurring anomaly. The circular suction cup scars, measuring roughly 2 to 3 centimeters in diameter and ringed with the sharp chitin teeth from the squid's tentacles, are not randomly distributed across the whale's body. They cluster. They concentrate heavily around the whale's lower jaw and the complex of nasal passages located in its massive head. This is not the pattern you would expect from a terrified animal thrashing blindly in the grip of a predator. A dying squid would leave marks wherever the whale's teeth and jaws made contact, across the torso, the flippers, the tail. The tight clustering around the jaw and the nasal complex suggests a different dynamic. The sperm whale's nose is not just for breathing. It contains the spermaceti organ, a complex structure of oil-filled sacs that the whale uses to generate powerful, focused acoustic clicks for echolocation. By forcing air through these sacs, the whale produces sound waves so loud they can stun prey, and so precise they can map the internal organs of a squid hiding in the black water. It is a biological sonar array, a sensory weapon. Some researchers have proposed a disputed interpretation of these clustered scars. They argue the squid might not be struggling to escape. They suggest the squid could be deliberately targeting the whale's sensory weapon during combat, wrapping its tentacles around the jaw and striking at the acoustic complex to disable the whale's ability to track it. This is a theory, not a proven fact. The mechanics of a giant squid engaging a 40-ton whale in tactical combat are almost impossible to verify without direct observation. But the clustering of the scars is a measurable, physical reality. It forces an unsettling question about the nature of these deep-sea battles. What if the squid was not just defending itself? What if the neural complexity, the advanced visual processing, the calculated strike capability meant the squid was actively hunting the whale? Some researchers argue that the expansion of neural genes implies a form of behavioral flexibility, perhaps even a form of consciousness or self-awareness that we refuse to attribute to an invertebrate. That does not prove the wildest version of the story, but it does make the simple version harder to defend. If the giant squid possesses a brain wired by the same genetic mechanisms that drive complex vertebrate intelligence, its behavior cannot be reduced to simple reflex. The encounters described in historical logs, the deliberate wrapping of tentacles around hulls, the coordinated attacks, suddenly shift from impossible mythology to disturbingly plausible biology. The Kraken of myth was not a giant mindless snake. It was a calculating, aware, intelligent predator. The DNA suggests the sailors were not hallucinating. They were just describing the behavior of an animal that science had not yet understood. The reversal is complete. The creature we thought we had explained is more alien, more advanced, and more dangerous than the textbooks ever allowed. And this brings us to the modern conflict. The reason this genetic discovery feels less like a solved puzzle and more like an unfolding crisis. We have finally read the code of the Kraken, and we have realized we are sharing the planet with an intelligence we do not comprehend. And at the exact moment we make this discovery, we are destroying its habitat. The deep ocean is no longer a remote, untouched refuge. It is the new frontier for industrial extraction. The International Seabed Authority, the United Nations body tasked with regulating ocean floor resources, is currently overseeing a rapid expansion of deep-sea mining permits. The primary target is the Clarion-Clipperton Zone, a vast abyssal plain stretching across the Pacific Ocean between Hawaii and Mexico. This area, reaching depths of 4,000 to 6,000 meters, is littered with polymetallic nodules, potato-sized rocks rich in cobalt and nickel. The demand for these specific metals has skyrocketed, driven almost entirely by the global manufacturing push for electric vehicle batteries. The International Seabed Authority has granted over 30 exploration contracts to various nations within this zone alone, but these contracts were issued before comprehensive baseline ecological surveys could be completed. Mining contractors are deploying massive prototype machines designed to drag across the seabed and harvest these nodules. These machines generate sediment plumes that do not just settle. Current models show the disturbed silt laden with heavy metals spreading laterally for hundreds of kilometers, along with noise and electromagnetic interference that could be devastating to creatures adapted to absolute silence and darkness. If the giant squid possesses the neural complexity the genome suggests, its sensory world is likely incredibly sophisticated. We do not know what industrial-scale seabed stripping does to a brain wired by hundreds of protocatarans. We do not know how a creature that processes low-light visual data at superhuman speeds reacts to the sudden obliteration of the sediment ecosystem it evolved to hunt in. The International Seabed Authority is making permitting decisions based on environmental impact assessments, but those assessments are built on the old assumption that the deep ocean is mostly empty, populated by simple, drifting organisms. The genome says otherwise. There is a deeper institutional tension here as well. The museums that hold the intact specimens, places like Te Papa in New Zealand, are hoarding irreplaceable biological data. The Te Papa colossal squid is the largest intact specimen ever captured. It sits in that basement tank, perfectly preserved, but its genetic secrets were incredibly difficult to extract because of the ammonia degradation. Critics have noticed that the race to sequence the genome took far too long, hampered by the physical reality of specimen preservation and the slow pace of institutional access. While museums carefully guard their treasures, the Clarion-Clipperton Zone is being carved up by contractors above them. The ethical question is no longer abstract. If the Kraken possesses a level of cognitive processing that rivals some vertebrates, if its neural architecture is a product of millions of years of intense evolutionary pressure to build a better deep-sea predator, then our interaction with it is no longer just a matter of marine biology. It is a matter of ethics. Are we hunting a highly complex intelligence? Are we driving an advanced mind into extinction before we even hear it think? The debate has never fully disappeared, but the genetic evidence has shifted the burden of proof. The mining companies and the navies are operating on the old assumption. The genome says otherwise. You are left standing in front of the glass. The basement lab is cold. The hum of the freezer units is the only sound. The colossal squid floats in its formalin tank, suspended in the blue chemical glow. Its massive tentacles are curled tight against its body. The rotating hooks are visible, sharp, and dormant. And then there is the eye. The dinner plate eye stares out through the thick glass, dense and unblinking. Before the genome was sequenced, that eye looked dead. It looked like a fleshy anomaly on a stupid creature that didn't know enough to stay out of a fishing net. Now, the eye looks different. It looks like a lens. It looks like the J-Show Dwanko of a supercomputer. It looks like something that was watching the black water long before we invented the submarines to go down and find it. The small museum label on the rim of the tank lists its species, its weight, the date it was pulled from the Ross Sea. It reads like a description of an object. It does not mention the protocatarans. It does not mention the jumping genes. It does not mention the neural explosion hidden inside the flesh. The label feels incomplete now, almost like a cage description for a captive intelligence that the curators do not fully understand. The dumb sea monster story has collapsed. But what has replaced it is not a clean answer. It is a darker, deeper question, floating there behind the glass, waiting in the dark.